the regulation of mitotic exit by the Anaphase Promoting Complex, also known as APCC. Cell division is one of the most fundamental biological processes, ensuring the accurate segregation of genetic material from one cell to two daughter cells. At the heart of this process lies mitosis, a tightly regulated sequence of events that orchestrates chromosome alignment, segregation, and the eventual partitioning of cellular contents. Among the crucial phases of mitosis, mitotic exit stands out as a decisive transition, marking the shift from chromosome separation to the establishment of two independent nuclei. This transition is governed by a complex interplay of molecular regulators, with the Anaphase Promoting Complex, or APCC, standing as a key orchestrator. The APCC is an E3 Ubiquitin Ligase that plays an indispensable role in targeting specific proteins for degradation via the ubiquitin proteasome system. By selectively tagging mitotic regulators such as Securin and Cyclins for destruction, the APCC ensures the timely progression of anaphase and the orderly exit from mitosis. The precise regulation of this process is critical. Errors in mitotic exit can lead to catastrophic consequences, including aneuploidy and oncogenesis. In this documentary, we delve into the molecular intricacies of APC/C-mediated mitotic exit, examining its regulation, key substrates, and broader biological significance. Drawing on extensive experimental evidence, case studies, and examples, we explore how the finely tuned activity of the APCC ensures the fidelity of cell division and its implications in health and disease. Mitotic exit is the culmination of mitosis, ensuring that once chromosomes have been separated, they are properly enclosed within newly forming nuclear envelopes. This transition involves the inactivation of mitotic kinases, the degradation of mitotic cyclins, and the re-establishment of interphase cell structures. The APCC is the master regulator of this phase, primarily through its ability to mark key mitotic proteins for degradation. The APCC operates as an E3 Ubiquitin Ligase, a multi-subunit protein complex that facilitates the transfer of ubiquitin molecules to specific substrates. Once polyubiquitinated, these proteins are recognized and degraded by the 26S proteasome. This degradation-based regulation ensures the irreversible progression of mitotic exit, preventing any backtracking or errors that could compromise genomic integrity. One of the most critical substrates of the APCC is Securin, an inhibitor of Separase. Separase is the protease responsible for cleaving cohesin, the protein complex that holds sister chromatids together. During metaphase, Securin binds to Separase, preventing premature chromosome separation. When the APCC is activated, it ubiquitinates Securin, targeting it for degradation. This releases Separase, allowing it to cleave cohesin and trigger the onset of anaphase. This event is crucial for proper chromosome segregation, as defects in this process can lead to aneuploidy and chromosomal instability, hallmarks of many cancers. Additionally, the APCC targets mitotic cyclins such as cyclin B, which are responsible for activating cyclin-dependent kinase 1. CDK1 drives the early stages of mitosis, phosphorylating numerous proteins to promote chromosome condensation and spindle formation. For mitotic exit to occur, CDK1 activity must be extinguished. The APCC accomplishes this by ubiquitinating cyclin B, leading to its proteasomal degradation. The inactivation of CDK1 allows phosphatases such as PP2A to dephosphorylate mitotic substrates, promoting the reassembly of the nuclear envelope and cytokinesis. The APCC is not constitutively active throughout the cell cycle. Its function is tightly regulated to ensure that protein degradation occurs at the right time. Two key co-activators, CDC20 and CDH1, determine the timing and specificity of APCC activity. During early mitosis, APCC is primarily activated by CDC20. However, the activation of APC/C circumflex CDC20 is tightly controlled by the spindle assembly checkpoint, a surveillance mechanism that ensures all chromosomes are properly attached to the mitotic spindle before anaphase onset. If even a single kinetochore remains unattached, the SAC generates a mitotic checkpoint complex, or MCC, which inhibits APC/C circumflex CDC20 and prevents premature anaphase entry. Once all kinetochores are correctly attached, the SAC is silenced, allowing APC/C circumflex CDC20 to target Securin and cyclin B for degradation. Following anaphase, the APCC switches its co-activator from CDC20 to CDH1. APC/C circumflex CDH1 remains active through late mitosis and into the G1 phase, targeting additional mitotic regulators for degradation to ensure complete mitotic exit. Unlike CDC20, which is degraded after anaphase, CDH1 is regulated by phosphorylation. CDK1 phosphorylates CDH1 during mitosis, preventing it from activating the APCC. As CDK1 activity declines due to cyclin B degradation, CDH1 becomes dephosphorylated and can bind to the APCC, extending its activity beyond mitosis. This dual co-activator system allows for temporal control over APCC function, ensuring that different sets of proteins are degraded at distinct phases of mitotic exit. The regulation of APCC activity is further fine-tuned by inhibitory proteins such as MI1, which suppresses APCC function during S and G2 phases to prevent premature mitotic progression. The importance of APCC in mitotic exit is underscored by numerous studies using genetic and biochemical approaches. One landmark study by Irniger et al. In the year 2000 demonstrated that yeast cells lacking APCC activity fail to degrade cyclin B, leading to mitotic arrest. Similarly, studies in human cells have shown that depletion of APCC subunits or its co-activators results in prolonged mitosis, chromosome misaggregation, and increased genomic instability. Mouse models have provided additional insights into the role of APCC in development and disease. For instance, knockout studies of CDC20 have revealed that embryos fail to progress beyond the two-cell stage due to defects in chromosome segregation, highlighting the essential role of APCC in early embryogenesis. Moreover, mutations in APCC components have been implicated in various cancers. Loss of APCC function leads to the accumulation of mitotic regulators, resulting in uncontrolled cell division and tumorigenesis. In cancer research, APCC dysregulation has been linked to aneuploidy, a common feature of many tumors. Studies in breast and colorectal cancer have identified mutations in APCC subunits that impair its ubiquitin ligase activity, leading to chromosomal instability. Conversely, hyperactivation of APCC can also be detrimental as it may drive excessive degradation of mitotic regulators, leading to defective cell cycle progression. Given its central role in cell cycle control, the APCC has emerged as a potential therapeutic target in cancer treatment. Several small molecule inhibitors of APCC have been developed, aiming to exploit the mitotic vulnerabilities of cancer cells. One promising approach involves targeting APC/C circumflex CDC20, which is frequently overexpressed in aggressive cancers. Inhibitors such as proTAME have been shown to block APCC activity, inducing mitotic arrest and apoptosis in cancer cells. Another therapeutic strategy involves modulating CDH1 activity. Since APC/C circumflex CDH1 promotes the degradation of mitotic regulators that are essential for proliferation, enhancing its activity could suppress tumor growth. Compounds that stabilize CDH1 have shown promise in preclinical models of glioblastoma and lung cancer. The Anaphase Promoting Complex, or APCC, is a master regulator of mitotic exit, ensuring the precise degradation of key mitotic proteins to facilitate chromosome segregation and cell cycle progression. Through its co-activators CDC20 and CDH1, the APCC orchestrates the timely destruction of Securin and Cyclins, preventing errors that could lead to aneuploidy and tumorigenesis. The regulation of APCC activity is tightly controlled by checkpoint mechanisms, safeguarding the integrity of cell division. Experimental studies, from yeast to human models, have demonstrated the indispensable role of APCC in maintaining genomic stability. Dysregulation of APCC activity is a hallmark of cancer, making it an attractive target for therapeutic intervention. Emerging inhibitors of APCC function offer potential new strategies for treating mitotic defects in cancer cells, paving the way for novel anti-cancer therapies. Understanding the APCC and its role in mitotic exit is not just a fundamental question in cell biology, but also a critical aspect of medical research with far-reaching implications for disease treatment and cellular engineering.